Hi, in this video I am going to talk about the bioprospecting of immunosuppressive agents. Immunosuppressive agents has the ability to reduce the immune activity. Actually, we need the immune activity to fight to diseases and things, but the immunosuppressive agents is doing the opposite. And sometimes it is what we needed, especially in case of organ transplantation and like treating graft versus host rejection and autoimmune diseases. In those cases, we need to suppress our immune system, and for that we need immunosuppressive agents. Immunosuppressive agents can be an induction drug or maintenance drug. Induction drugs are actually used immediately after the organ transplantation. It's a drug to suppress the immune activity in a acute manner. And maintenance drugs are more chronic in nature, which is used in later stages to maintain the immune activity under control. So these immunosuppressant drugs come in different classes like glucocorticoids, calcineurin inhibitors, anti-proliferative or anti-metabolic agents, or biological like antibiotics. So the bioprospecting including the finding this immunosuppressive drugs from nature, natural sources like just like the plant and bacteria or even animals. But the thing is that we have seen several different types of bioprospecting so far. The enzymes, prospecting for the enzymes, antibiotics, or even for the uh, other compounds. But the thing is that immunosuppressive agents are very difficult to find from nature because the immune system won't function in cultured cells or cultured organs. It functions only in the whole organism. We cannot culture the cells and say that the whole immune system is there because it's like an organ. It need to be in the organism. We can actually study the immunosuppression in a whole animal, not in individual cells. But there are methods we can actually find to screen the immunosuppressive agents. So the methods we are using for screening for immunosuppressive agents will be different than the other methods. So our immune reaction can be of different types. We have the cell-mediated immunity and humoral immunity. Cell-mediated immunity, one of the main component is T cell. So first method of screening is using T cell activation assay. So for any immunological reaction, the one of the major step is the T cell activation. You might have heard about this T cell. This T cell activation involving many changes in the T cell, which can mediate long-term antigen-specific effect. The memory responses, the T cells will be converted into effector T cells and memory T cells, things like that. So some of the immunosuppressive agents, what they are doing, they are actually reducing the activity of T cell activation. They can prevent the T cell activation. If any natural compound is able to prevent T cell activation, it has a potential to be used as a immunosuppressive agents. So first method is T cell activation assay. In T cell activation assay, what we will do, we will take the whole blood and then we'll separate the components. From that we will select the T cells. And we will have three different conditions. One is control. In control, we are adding only T cells there. Then second one is T cell activation. We are adding compounds to activate the T cells. So some like interleukins or TNF alpha compounds like that will be added to the medium to activate the T cells. And in the final set, we are adding T cell activation along with the compound which has the potential anti like immunosuppressive activity. So if the compound is having immunosuppressive activity, the compound will prevent the T cell activation even in the presence of T cell activating chemicals. So if any compounds is found to be like prevent the T cell activation, it may have a potential to be used as a immunosuppressive agents. That's one way. Then the humoral part of cell immunity is handled by the B cells. So we have a B cell proliferation assay. It is done the same way as the T cell assay. Here we isolate the B cells and we will add the compounds to induce the proliferation of B cells and maturation of the B cells. If our compound is able to prevent that maturation and proliferation, this compound have a potential immunosuppressive ability. So this is the pathway of B cell maturation. So if you are adding some chemicals is able to prevent the conversion of the mature B cells into plasma cells and memory cells, that compound can be used as a immunosuppressive agent. And as I said before, immunosuppression can be studied only in the whole animal. So most of the immunosuppression screening is done in experimental animals. And mostly we are using mouse and rat, and we can actually study the immunotoxicity of the compounds in these animals. And we are using tier testing system. It has multiple tiers, the first tier and second tier. In the first tier, for any compound, we are actually testing the immunotoxicity. The first thing we want to know whether the compound is toxic, and if it is toxic, we will stop our experiment, and if it is not toxic, we will go to the second tier. In the second tier, we will have a more detailed study to see that whether the compound is actually having an immunosuppressive capacity. So for the screening of immunosuppression, first we will do T cell assay or B cell assay. If the compound has some activity, we will take it to the animal experiment. Sometimes even without the doing this assay, we can actually take the compound directly into the animal experiment. So here what we do here, this is a transplantation experiment. We are taking liver from this donor animal to this recipient animal. Once the liver is here, we will give the immunosuppressants, the potential drugs to the rat. If the potential drugs are effective, it will prevent the graft rejection. Okay, so if it is not effective, the graft rejection will be there, this animal will die. So we will do the effectiveness studies in the liver cells to see that whether there is any immunological reaction. If our compound is good, it won't happen. It won't reject the transplant. So many immunosuppressive agents has been discovered from nature, which including cystosporin, which is found from a wild fungi, and it is used for the treatment of rheumatoid arthritis, Crohn's disease, and organ transplantation to prevent rejection, to work by decreasing the functioning of lymphocyte. Then colchicine is an anti-inflammatory agent, which is also found from a plant. It's used for the treatment of gout. Other than this immunosuppressive agents, we can use the bioprospecting food to find other metabolites, like therapeutically useful metabolites like vitamin B12. It's a metabolite which is used as a treatment for diseases, or the some other vitamins producing microorganisms. Microorganisms can actually produce these vitamins. So this screening outline is given here. In here, what we are doing, we are taking samples from different sources like soil, water, and air, and it is diluted. And once we diluted, we are actually plating it in a medium which lack the metabolite we are going to need. So if you are looking for an organism which is producing vitamin B12, we are plating the cells, the samples in a medium which lack vitamin B12. So the only organism which is surviving in that medium is the one those can produce the vitamin B12. If the organism does not have the ability to produce vitamin B12, they cannot actually grow in a medium which lack vitamin B12 if it is an essential nutrient for the organism. And then what we do, after incubation, we are incubating it with a noxotrophic organism, which can grow only in the presence of particular compound. So if our sample containing like microorganism which is producing the essential nutrient, this microorganism will grow and produce the essential nutrient and release into the surrounding medium. Okay, and in the surrounding medium, what we have the another microorganism which need that nutrient. If the indicator organism or test organism is growing around that colony, which means that this organism at the center of this colony is producing some metabolite useful for the test organism. So in this test, we are using a test organism. Okay, the test organism can be only grow in the presence of the particular nutrient. Okay, this particular nutrient is lacking in the agar medium. So in case of vitamin B12, if you are looking for a organism which is producing vitamin B12, first we need to make a medium which lack vitamin B12. Then take samples from different natural sources, plate it over it. Okay, after plating, we apply test organism. The text organisms are oxotrophic in nature, which cannot produce vitamin B12 and which need vitamin B12 for the growth. So medium is lacking the vitamin B12. In this medium, actually, this microorganism do not grow. But any of the microorganism derived from the soil, water, or air sample is producing vitamin B12 and giving into this test organism, it will grow. So the growth of test organism will be around the colonies, which is actually producing this particular nutrient. That way we can actually find the metabolite producing microorganism. Another example is organic acids. Organic acids like citric acid is used extensively in the food industry. So the organic acid producing microorganism can be also screened from the natural sources. So for that we are actually taking samples from soils, water and air, then diluted to get isolated colonies, and these colonies are plated in a medium which contain a pH indicator. If the change of the if the pH if there is a change in pH of the medium, it will change color. So our agar plate is having a pH indicator. So if the microorganism is producing an acid or organic acid or any type of acid, there will be color change in the medium. So the colonies which is producing the acids can be easily identified after incubation, because the colonies producing acid will have a color change around their colonies. Okay, in that way we can actually find the microorganism very easily. That's the methods of screening used. That's bioprospecting methods we are using. Thank you so much for listening.